Now students, we are going to study a third receptor family, that is G protein coupled receptor. As clear from the word G protein coupled receptor, it means these are the types of receptors which are actually linked with a protein, and that protein is known as G protein, and this receptor is a transmembrane receptor. So this receptor consists of two domains. First is extracellular domain and second one is intracellular domain. Extracellular domains contains ligand binding site, and the intercellular domain interacts with the G protein after activation. Now, types of G protein. There are three types of G protein coupled receptor. First one is the receptor which is linked with the GS protein. Second receptor is linked with GI protein. GS is stimulatory protein and I is inhibitory protein. Now third type of G protein coupled receptor is linked with GQ protein. Each of these G proteins consists of three subunits: alpha, beta and gamma. This alpha subunit actually binds GDP, that is guanosine diphosphate, when it is in its inactivated state. When it activates, then there is binding of GDP with the alpha subunit. Then the second subunit is beta subunit, and the third subunit is gamma subunit, which actually anchors the protein in this lipid bilayer or the cell membrane. Now, when the receptor is in its inactivated state, that means there is no binding of ligand, hormone or neurotransmitter to the receptor. It means there is no interaction of the protein, there is no interaction of the receptor with the protein. However, whenever there is activation of the receptor, whenever there is binding of the ligand, agonist, neurotransmitter or hormone to the receptor, there is activation of the receptor. As a result of this activation, there is association or there is link between this G protein and this receptor. Now, after activation, there will be formation of a linkage between this G protein and this receptor. Afterwards, there will be replacement or dissociation of the GDP with the GTP. Now, this GTP with this alpha subunit will make a complex, and this complex will move towards the effector molecule. Effector molecule may be an enzyme, protein or ion channel. In this figure, the effector molecule is an enzyme, that is adenyl cyclase. So after the interaction of this GTP, this protein with the effector molecule, there will be a biological response. Now, depending upon the type of the protein, there will be the biological response. If this is a GS protein, that is stimulatory protein, as a result of this interaction, there will be activation of the receptor, that is adenyl cyclase. If this protein is GI protein, there will be inactivation of the receptor. Now this complex is actually binding or interacting with the enzyme, that is adenyl cyclase. So the response usually lasts for second to minutes in the case of G protein coupled receptor. The activated effectors, that is enzyme in this case, so the activated effector will produce a second messenger. In our case, the activated effector is producing a second messenger, that is cyclic AMP. Now moving towards the figure, whenever there is binding of a neurotransmitter with a G protein coupled receptor, in this figure, our neurotransmitter is norepinephrine. Whenever there is binding of norepinephrine with beta receptors, that are G protein coupled receptor with beta 1 or beta 2 receptor, which are GS proteins, which are linked to GS protein, there is activation of the effector, and that effector is adenyl cyclase. As an activation of this effector, as an activation of this enzyme, there is formation of cycle IAMP, there is formation of second messenger from ATP, and then as a result of this second messenger, there will be a biological response, and in the case of this cardiac muscle, in the case of this cardiac cell, the response is the contraction. Now, A common effector activated by GS and inhibited by GI is the adenyl cyclase, which is an enzyme which produces a second messenger, and this second messenger is cyclic AMP, that is adenosine monophosphate, which is formed from ATP, that is adenosine triphosphate. Again, this figure is representing example of G protein coupled receptor, and these examples are alpha and beta adrenoreceptors. As a result of activation of these receptors, there is formation of second messenger, there is formation of cyclic AMP, there is protein phosphorylation. Now, whenever there is formation of cyclic AMP, there is activation of an enzyme, that is protein kinase A, and as a result of activation of this enzyme, there is phosphorylation of proteins. As a result, there is a biological response within the cell. Now, moving towards GQ coupled receptors. Whenever your receptor is linked or associated with GQ protein, it means whenever there is activation of GQ protein, whenever there is activation of the receptor, there is actually activation of an effector molecule, there is activation of an enzyme, that is phospholipase C. As a result of this activation, there will be formation of two second messengers. First one is IP3, that is anisotol 1,4,5 triphosphate, and DAG, that is diacylglycerol. So these two are actually the second messengers. And these two second messenger will result in activation of a protein kinase, that is protein kinase C. Now, additionally, these two messengers have ability to cause calcium influx and release calcium within the cell. This DAG will result in influx of calcium through a calcium channel from outside of the cell to the inside of the cell. And this IP3 will then release the cytosolic or the calcium which is present in the endoplasmic reticulum, and this endoplasmic reticulum is in the cytosol. As a result of formation of this IP3, there will be release of the calcium from the endoplasmic reticulum. And this IP3 will also facilitate DAG in the influx of calcium ions. So as a result of this calcium, as a result of increased content of this calcium in the cell, there will be contraction within the cell. Now, this figure is representing that activation of GQ protein will result in activation of phospholipase C. As a result, there is formation of two second messengers, DAG and IP3, and these will result in activation of protein kinases and influx or release of the calcium. As a result, there will be protein phosphorylation or there will be contraction. Again, this figure is representing that this blue area is extracellular fluid. Here we have a protein that is linked with GQ protein. Then in the cell, which is represented in pink color, we have endoplasmic reticulum, which is containing calcium. Whenever there is activation of GQ protein, then GQ protein will link itself with GTP. As a result of this activation, as a result of this association, there will be activation of an enzyme, that is phospholipase C. This phospholipase C is actually producing second messengers from phosphatidyl inositol bisphosphate. Okay, PIP2 bis, this 2. Okay. Now from this PIP2, there is formation of DAG and IP3, which are the second messengers. This IP3 is now acting or working with the endoplasmic reticulum. It is working on the calcium channels of the endoplasmic reticulum. It is working on IP3 gated calcium channels. As a result of this, there will be activation of the calcium channel. There will be release of calcium from the endoplasmic reticulum into the cytosol. Now this calcium is released in the cytosol. Secondly, this tag will result in activation of phosphokinase C, which is an enzyme, and this enzyme will cause phosphorylation of the substrates or proteins. Additionally, this will cause the opening of the calcium channels which are present on the cells, As a result of this calcium channel opening, there will be release of calcium into the cytosol. So this calcium is released by IP3 as well. Calcium released by IP3 is to work with DAG to activate protein kinase C. So both of these are working together in releasing calcium and the contraction of the cells.